Okay, good afternoon, everyone. A very warm welcome to which hopefully will be the second and last annual CGU online research presentations. We did a version of this last summer, as some of you will remember. So this is going to continue running through June and July, typically on Tuesdays and a few Thursdays throughout the summer. We're basically looking at our general hydrology session today with a theme of ecohydrology with lots of wetland content. So hopefully there's lots of kind of wetlanders out there. I'm joining you today from Brandon University in Brandon, Manitoba, and would like to note that we respect the treaties that were made on these lands and acknowledge that Brandon University is located on Treaty 2 lands. So on behalf of Brandon University, I welcome you to the traditional homelands of the Dakota, Anishinaabe, Ojibwe, Cray, Dene, and Métis people. And I encourage you to know your land acknowledgements for where you're coming from today across this country, or if you're international. So before we start with our speakers, we're going to display a short video at the end of the meeting for hopefully what will be an in-person meeting for next year, themed Science Serving Society. So please do continue to the end, and so that we can watch that together. We'll also add the YouTube link to the chat. Just some logistics for those of you that are joining as attendees. We can unmute you if you would like to ask a question verbally as opposed to typing it, or you can just type it out in the Q&A. So there's at the bottom, you're probably familiar with Zoom by now, but with chat, but there's also a Q&A box, so you can post your questions in there. But if you prefer to speak your question, then you can just let us know, and we can accommodate that. So our first speaker today, I'm just pulling up this session, is Sheryl, coming from the University of Waterloo, talking about upscaling spatial and seasonal evapotranspiration using in situ measurements and high resolution imagery in a subalpine meadow in the Canadian Rocky Mountains. So I'll just remind our speakers that our talks are 15 minutes with a few minutes for questions. So if you're kind of coming in and around the 12 or 13 minute mark, that would be great. I believe that we will be prompting either through a chat or a pop-up of some description if you're coming close to your time limit. So if you're not able to pay attention to the time, or depending on your screen setup, if that doesn't work for you. So I believe Sheryl, you just start, you turn on your video and share your screen, I believe, and then we go from there. And I'm going to mute myself. Do you have any questions, Sheryl, before we start? Or are you good?
No, that should be good. Thank you. Oh, I think it says host disabled participant screen sharing. So I need to, oh, there we go. Never mind, it worked.
Yeah, perfect.
Okay, everyone see that okay?
Well, if that isn't a spectacular picture, I don't know what is. So I'm looking forward to this.
Yeah, it's a gorgeous site, that's for sure. Yeah, so thanks, Pete, for the introduction. Yeah, just as he said, my name's Sheryl, and I'm a master's student at the University of Waterloo. So yeah, today I'll be presenting a talk about upscaling spatial and seasonal evapotranspiration in a subalpine meadow in the Canadian Rocky Mountains. This project was conducted with the help of my supervisor, Richard Patrone, and my co-authors, Philip Harder and John Pomeroy. First, I'd like to give land acknowledgement as well to the Blackfoot Confederacy, the Tsuut'ina, the Stoney Nakoda Nations, and the Métis Nation in Alberta. I would also like to thank our sponsors for their financial contribution to the project. And last but not least, thank you to my field partners, lab technicians, the field crew at the Coldwater Lab in Canmore, and everyone at Fortress Mountain for a great field season. I'd like to start off with some background information about evapotranspiration in subalpine ecosystems and its importance to local hydrology. So mountain ecosystems supply water to downstream communities for residential, agricultural, and ecological function. As temperatures rise, snowmelt patterns are shifting to earlier spring melt and later refreezing times, increasing the probability of flooding of downstream communities. Subalpine wetlands function as buffers for water flow and are important ecosystem for regulating water quantity. Evapotranspiration, or ET, is an important output in the water balance of these ecosystems, which is likely going to change with warming trends. These ecosystems are especially vulnerable to climate change, as models show highest rates of warming in high elevation regions. This could further invite species migration of lower elevation plants due to warmer climates, longer and earlier growing seasons, which puts subalpine plant species diversity at risk. As plants have varying rates of ET, water fluxes could change and further influence wetland buffering ability. ET is sensitive to hydrometeorological conditions, including net radiation, temperature, and moisture regimes. In addition to warming, climate models predict increased variability in precipitation. Mountain ecosystems are also sensitive to terrain shading dynamics due to large elevational gradients in the landscape, which further influence microclimatic conditions and ET response. It's not well studied how shading dynamics in mountainous regions reduce ET for plant communities that could be at risk to species migration, and how their evaporative function contributes to ecosystem ET. This is often due to difficult accessibility and limited spatial resolution of available clear sky satellite imagery, which is often too coarse to detect change at the plot scale. Therefore, it's important to recognize these communities' contribution to ecosystem ET for environmental monitoring of subalpine wetlands and their hydrologic function. So the objectives for this study are to quantify the spatial and seasonal variability of ET at the plant community scale across a subalpine meadow, and to estimate upscaling parameters for ET at the plot scale with high resolution UAV imagery to determine the relative contribution to ecosystem ET. On the right there's some examples of individual plots of plant communities showing a lot of species diversity at the site. So this was done on Fortress Mountain in Kananaskis, Alberta, at a temporary subalpine meadow commonly referred to as bonsai. The meadow is at an elevation of 2,090 meters, and the study area is just under 5 kilometers squared. Temporary meadows are characterized by mineral soils that are drier than typical wetlands for most of the growing season, but they retain enough water for vegetation to grow after flooding events caused by snowmelt or periods of heavy rainfall during the growing season. As a result, the meadow is home to a variety of subalpine plant species, with the most dominant vegetation types being horsetail, fleabane, various types of willows, larch trees, and balsam firs. There's also quite a bit of brown moss along the stream. As the site is heterogeneous and home to diverse species, the plant community types were categorized into 3 groups. So we have the trees, including the larches and firs, the shrubs containing all the willow species, and ground vegetation, which encompasses all shorter vegetation types, including horsetail, moss, fleabane, and other wildflowers. These community plots are scattered throughout the site, and point measurements of ET were collected using a dynamic closed chamber. There's also a meteorological station near the middle of the study area. The station collects eddy covariance data, including ecosystem ET, and hydrometeorological data, such as air temperature, relative humidity, soil moisture, and soil temperature. There's a second meteorological station about 100 meters north of the study area that collects precipitation data, as well as 4 surface temperature sensors across the site. So typically the growing season at bonsai starts in early to mid-July to early September. In this project, we'll just look at the tail end of the growing season from August 24th to September 11th, 2020. So during the study period, the average daily temperature stays around 10 to 12 degrees Celsius in the first few days, then continues to drop down to 5 degrees at the end of the week, and it coincides well with the daily ecosystem ET, which starts off at 2 millimeters per day and drops to 0.25 millimeters per day in the same time frame. You can also see a decreasing trend in all plant community groups on the 3rd graph, although these are only point measurements of ET, which have been temporally upscaled to hourly measurements. They're also normalized by LAI for comparison of change over time. In the 2nd week, temperatures rise back up to 12 degrees and drops to freezing temperatures at the end of the week, with 7 millimeters of snowfall on September 7th. Again, the ET measurements reflect this trend with a decrease in temperature and ET on September 3rd and 7th. After the first snowfall in the late growing season on September 7th, ET remains relatively low, which is to be expected as some plant communities are not frost resistant and they die off at the end of the season. Plants that are frost resistant likely aren't transpiring due to residual snow cover. As expected in the two graphs on the right, ET decreases at the end of the tail end of the growing season at the ecosystem level and in all plant communities. To see how these communities respond to shading, we'll look at some of the variation in ET measurements during sunny and shaded measurements. Looking at a clear sunny day last summer on August 26th, in the video, the site is just below the talus slope, and you can see varying levels of terrain shading throughout the site. The video is in increments of 3 hours, starting from 6 a.m. to 9 p.m. And on the southeast part of the meadow, on the left side of the video, it gets shaded first in the morning, and the northwest part on the right side of the video gets shaded first in the late afternoon. Looking at the two graphs on the right with the ecosystem ET on top and net radiation on the bottom, we can see a big dip in net radiation from 1 to 2 p.m. Ecosystem ET decreases at the same time, suggesting that shade has an impact on ET at the ecosystem scale, even within a very short time frame. To the left, you can see how terrain shading affects the site. The camera is pointing east, and you can see uneven terrain shading at this point. Within the half hour, the whole site will be shaded. To the right, we have normalized plot scale ET measurements over time, separated by sunny and shaded measurements. The sunny measurements were consistently higher than the shaded measurements. And to look at how shade affects different plant communities, we'll focus on 2 measurement days before the snowfall event on September 2nd, or September 7th, sorry. So those 2 days are September 2nd and September 5th. On September 2nd, measurements were taken from 12:30 to 2:30 p.m. And on September 5th, measurements were taken from 11:15 to 12:45 p.m. These 2 days vary the most in temperature and radiation, but overall they have a similar soil moisture and daily ET rate at the ecosystem level. At the plot scale, we can see variation of ET in all plant communities in both shaded and sunny measurement days. Again, these ET rates are normalized by LAI, and Moss LAI was estimated using regression. As expected, plant community ET is higher in all the sunny measurements, although some plants seem to be more sensitive than others, with shrubs showing the highest difference between sunny and shaded measurements. Mosses also show quite a bit of evaporation in the sunny days. In the classification map, you can see the diversity of plant communities over the study site. There's a large variability of species composition at the site with varying plant community ET, especially in the sunny measurements before snowfall. Again, between plant communities, there's a high variability of ET between the communities. Overall, shrubs had the highest ET per LAI at the site on sunny days, and firs had the lowest. As firs are the only evergreen species, it's likely due to stomatal control and wax coating on the fir needles. The shrubs also had the highest sensitivity to shade in all of the measurements. And as plant communities vary spatially over land cover, it's important to see their relative contribution to ecosystem ET. So there's also a high spatial variability in hydrometeorological and vegetation parameters often used for ET modeling due to heterogeneous land cover, species composition, and terrain shading dynamics. On the first map on the top left corner is the soil moisture, map A. And you can see quite a bit of variability in soil moisture, which is likely due to differences in surficial geology. There's quite a bit of rock gardens. Again, it's at the end of the talus slope. And the map right next to it on the top right shows surface temperature. And there's quite a lot of variability in this as well, which is also likely due to land cover differences in albedo and heat absorption. And then the bottom two shows NDVI, so vegetation health, and LAI, so leaf area index. Increases towards the stream and water resources, as well as larger plants, can be seen pretty clearly here. And so to wrap this up, plant community ET varies in response to changes in the microclimate, including shading dynamics. ET varies across the study site and between plant communities, which makes it difficult to capture ET spatially in heterogeneous ecosystems such as bonsai. However, high-resolution imagery can be used to estimate variability at the plot scale for parameters in ET modeling. And further research in this project will use these estimates for ET upscaling from the plot scale to the ecosystem scale. And that's about it. Thank you.
Excellent, Sheryl. Thank you very much. I don't see any questions yet, but I have a fairly simple question, I think. If you wanted to go back to your study area map, I'm just curious. So you said the study area was roughly 5 square kilometers, and it looked like it was a square. How did you pick the boundaries for your square? I mean, a lot of people sort of pick a watershed or some other boundary that might have more intrinsic meaning, but I'm just curious. Presumably the black line is your study area.
Yeah, so this was actually, it was derived basically based on data availability. So we mostly for the soil moisture measurements, actually, because soil moisture was collected at the same time as the chamber measurements through a handheld moisture probe. And so it was basically based on the interpolation area that we could get from that.
Oh, okay.
That makes sense, yeah.
And so don't see any other questions as well, but I'm just curious. So reasonably, like a lowish elevation, how far do you have to walk or drive? Like I'm just curious of the logistics of working in the mountains, basically. Like how far are you from a vehicle? Like if you're transporting heavy equipment, is it, I'm just, yeah.
Yeah, it's definitely a little bit of a struggle. But yeah, so essentially it's just south of the Kananaskis village. I'm not sure if you're familiar with the area, just off Highway 40. So you can actually drive quite a bit of ways up to kind of like a side road at Fortress. And typically, if it's not too muddy, you're able to drive almost directly to the site. However, that road is kind of, I don't know, it's getting a little bit sketchy nowadays. So you're kind of walking, you do have to walk not too far. It's about, I don't know, I want to say like 2 kilometers maybe along a dirt path, but it's not too bad. Yeah, it's just bad when you're carrying heavy equipment. So the thing with the drones is it's quite nice because you can actually, because they have a good enough battery life to be able to fly from the road and then still capture the site from there. So
excellent. Yeah. Well, I'll stop bombarding you. I don't see any other questions in the Q&A area. I'll just acknowledge also that April has a question. April, if you want one of my other co-conveners with April and with Andrew as well.
Yeah, do we have time?
I think we got about a minute. Yep.
Okay, I just wanted to ask, thank you, Sheryl. I wanted to ask a little bit more about the measurement techniques you were using for data collection.
Mm-hmm.
Yeah, I mean, it was a pretty general question, but I wondered if it overlapped. I wonder if it overlapped with other studies, or was this, were you able to complement other groups that were doing data collection? It looks pretty great data set.
Oh, sorry to cut you off there. Yeah, actually the the master's student before me who worked with Rich, his name's Dylan Harach, he actually did his thesis was based on the shading dynamics at this site throughout the entire growing season. That was originally the plan, I guess, for mine with the plant communities, kind of going down to a more plot scale, but throughout the whole growing season. But as I'm sure other students were affected by it, we weren't really able to get out that early due to COVID. But yeah, there's actually been a lot of research at Fortress Mountain, primarily by John Pomeroy and Masaki Hayashi at Bonsai. So it is kind of an addition to those kinds of projects. Does that answer your question?
Yeah, great, thank you.
Excellent. So we'll switch over to our next speaker. So Sheryl, if you can mute and turn off your video, and then we'll get Brandon to do the opposite. And so Brandon will be assessing the potential change in the timing of thaw and ice-free conditions in a boreal peatland with climate change, also coming to us from the University of Waterloo. So looking forward to this. You'll be able to get your screen shared. Yep, there we go.
All good?
Yeah, I can see that too.
You can hear me?
Is that Scotty Creek?
That is not Scotty Creek. That is
No.
Fort McMurray, Passiflora.
Fair enough. Okay.
So yeah, as mentioned, my name is Brandon Van Huizen. I'm a PhD student under the supervision of Dr. Rich Patron as well, similar to Sheryl. And I'm presenting on some of my PhD work here, looking at freeze-thaw cycles in a boreal peatland. So to start, I just want to acknowledge all of the different funders, as well as acknowledging that this research was conducted on the traditional territories of the Cree, Métis, and Denendeh peoples, as well as a big thank you to the men listed there who helped me in the field, particularly for this portion of the study. So peatlands, as you may or may not know, are important stores of carbon and water in the Western Boreal Plains, and they're able to persist despite the presence of a subhumid climate. So potential evapotranspiration often exceeds precipitation on an annual basis. And they're able to store all of this carbon and water because you have these thick organic soil layers that are highly porous. And so up to 96% porosity in many cases in the upper layers. And because of this, they're very wet and decomposition rates are very slow. And so a lot of organic layers able to build up over time. And so these peatlands are important sources of water in the landscape. Sometimes they are sinks of water in the landscape. And in particular, the spring thaw represents an important hydrological period for peatlands in this environment. So the image on the left shows spring melt in a peatland. That's immediately after snow melt, and you can see there's lots of ponded water sitting at the surface there. And part of the reason that there's so much ponded water is because of the presence of seasonal ground ice. So this is not permafrost. This is ice that forms in the winter and then melts in the subsequent spring. And you can see an image of a core we've taken in one of the springs at my field site. And you can see it's a thick concrete layer that can be anywhere from 5 to 30 centimeters thick, sometimes even thicker. It all depends on sort of overwinter processes. And because it's so thick and relatively impervious, there's not a lot of infiltration capacity for the peatlands. And so when the snow melts, it has nowhere to go but run off of the peatland or evaporate. And so it has a large influence on the spring freshet and whether a peatland can act as a source of water within the landscape, particularly in headwater catchments in the Western Boreal Plains, where they typically act as sources for down gradient systems. But as many of us are probably aware, our climate has been shifting for some time and it is projected to continue to shift. So these are plots from the Canada's Changing Climate Report released back in 2019, zoomed in on Alberta. And we know that basically air temperatures have been increasing in the past and are expected to continue to increase in the future. And this has implications for ground temperatures. So we know that ground temperature, so the surface temperature is all likely to increase, but it's not necessarily clear by how much. And ground temperature is ultimately what initiates that freezing process within a peatland. You need that temperature to drop below 0 for freezing to start. And so if our ground temperatures are going to increase, there's a question as to sort of the length of the freezing season, as well as just how much ice can form and where it will form, but also what impact this might have on peatland freeze-thaw cycles. So is the ice going to persist for the entire winter, or is it going to have thaw periods throughout the winter as the climate warms? And so the objectives of this study were to conduct a sensitivity analysis to understand the impact of varying surface temperature on future freeze-thaw conditions, and then also to quantify the number of freeze-thaw cycles under each sort of sensitivity warming scenario. So this study took place, as mentioned, not at Scotty Creek, but at Passiflora Fen. It's a poor fen located about 40 kilometers south of Fort McMurray on Stony Mountain, which is sort of a topographically high point in the area. It's about 200 meters or so above the surrounding landscape, and it's located in a headwater catchment. And so at this site, we installed thermocouple profiles, as shown on the image on the bottom left. That little white box there is the logger box for the thermocouple profile. We installed it in sort of the central portion of this particular peatland. And so to conduct the sensitivity analysis, we used observed ground temperature data from this logger box. So it measured temperatures at 2 centimeters, 5 centimeters, incremented all the way down to 150 centimeters. And the organic soils here are very, very thick at this particular area where our thermocouple profile was installed. Average peat depth was about 4 meters. So pretty deep peatland. So to conduct the actual sensitivity analysis, we first used the Stephan's equation to model freeze thaw in the peatland. So the Stephan's equation is a relatively simple temperature index model, again, using that upper surface level temperature that was measured at 2 centimeters. And then it uses that temperature index in conjunction with physical properties to calculate the soil to calculate a thermal conductivity, which sort of dictates the efficiency at which energy is transferred through the soil and how deep your freezing in that soil can get. And so to drive this model, as I mentioned, we use that 2 centimeter thermocouple layer. And then to conduct the different sensitivities to it, alterations, I should say, we used a percentage increment approach where, say you had a temperature on January 1st that was, let's say, -2 degrees measured at the top of the surface. Then we took that value and added to it a certain percentage of the mean of whatever month that value happened to be in. So if it was January 1st, we took the mean January temperature across, and that means based off of our entire data set, so 2012 to 2019, and then added either 10%, 20%, 30%, et cetera, to that daily observed value. And we did that for every single day. And that's how we sort of conducted this sensitivity warming analysis. And then as far as the physical properties go, we assumed a porosity of 0.96. That's based off of soil cores taken at this particular location. It has very loose, fluffy moss and peat for the majority of the top 50 centimeters. And then also we assumed frozen conditions, saturated conditions at the time of freezing. So that peatland was completely filled up with water when freezing would begin. And so we ran this model and sort of averaged the results across all of the years and specifically looked at the timing of thaw and the timing of ice-free conditions. So this first result here is showing the average timing of ice-free conditions. So on the x-axis, we have average day of year, 0 being January 1st. On the y-axis, we have the different model runs. So the base model run is just using the observed temperature, no changes made to the model. And then each subsequent model run is below it. And then on the right-hand side, you can see those numbers descending down. That represents the difference between each model run and the base model run. In this case, the difference in the timing of ice-free conditions. And so what this is showing is that for the first 3 model runs, there's not that much of a shift in the timing. You get on average 2 to 3 to 5 days earlier. So, not quite a week. However, once you reach 40%, there's a market shift, and that number jumps up to 14 days or earlier on average. And then again, sort of a steady shift over time up to almost 2.5 weeks earlier. For the timing of thaw, we see a very similar trend again, and to the timing of ice-free conditions, in that around that 30 to 40% mark is where there is a market difference. And the reason for that difference is this represents sort of the threshold at which temperatures start to go above 0 degrees more frequently. And that's why there's such a big marked jump there. But the timing of thaw does shift between 30 and 40% from about 4 days earlier to 17 days earlier, and increases all the way up to about 25 days earlier. But you might be thinking, okay, these are just sensitivity results. How do we sort of contextualize that with climate change projections? So to do that, we took a look at data from Thompson et al. 2017, where they ran an ensemble of climate models and got mean air temperature projections for the Western Boreal Plains. And they ran it for climate projections in 2020s, the 2050s, and the 2080s. And so if we know what the projected air temperatures are going to be, we then use data from our on-site MET station and developed a relationship between that ground temperature value at 2 centimeters and the air temperature above it. And using that regression equation, we can then convert the graph on the left from air temperature to projected ground temperatures. Again, we only do it for April to October because, as shown in that previous figure, that relationship is only valid for when temperatures are above 0. And that's because during the winter time, there's actually a bit of a decoupling between the ground surface temperature and the air temperature above, usually due to the presence of some sort of snowpack that can be quite insulating. And so the variability in air temperature over the coldest portions of the winter is not really reflected in the ground temperature below. But now that we have average ground temperature, we can sort of start to fit in our sensitivity analysis. And to do that, we focused in specifically on the month of May, because that's when all of the melting typically was occurring. And so you get a graph like this. So the dots represent the mean May temperature for ground temperature for each of our sensitivity runs. And then the hash marks represent the projected mean May temperatures based off of that regression equation. And depending on how close each of the model runs fall to that mean sort of climate projection, gives us some degree of confidence in sort of suggesting, just how quickly is these shifts in the ice-free season, how quickly are they going to occur? So using that graph, I just overlaid it again with the first graph for the timing of ice-free conditions. You can see by the 2020s, it looks like we get into that 30% range. Maybe there will only be a shift of about 5 days, but once you get to the 2050s, that's where we see, potentially could see that sudden shift of up to 2 weeks earlier. And then by the 2080s and the end of the century, this suggests that, we could potentially see a shift up to 16 or 17 days earlier in the timing of ice-free conditions. And we can do the same thing with the timing of thaw, again, using that exact same approach. You can see by the 2020s, it suggests that, we might reach that 4-day on average earlier timing of thaw. And then again, by the 2050s, sometime around there, it's suggesting that we could see that more market shift. And then again, by the end of the century, could be seeing a shift from about the end of April to mid-April. So what does this mean for freeze-thaw cycles? Well, again, using that exact same approach, we counted up the number of freeze-thaw cycles and averaged it across all the years that we had. And you can see that—
Just about a minute left, Brandon.
Okay, you can see that there's a pretty steady increase. And basically, the number of freeze-thaw cycles are expected to get up to, or it suggests that the number of freeze-thaw cycles could be around 4 times on average by the end of the century. So what does this all mean? Well, as the climate warms, the timing of ice-free conditions are going to shift earlier in the season. And again, that's not necessarily surprising. But what that means is that the role for a peatland in the landscape may change, where spring discharge may become more variable, especially for headwater catchments. So more water is potentially going to be staying in the system as opposed to being shedded to down gradient systems. And the number of freeze-thaw cycles will also likely increase on average. And this has implications for late winter carbon fluxes. Typically, you have carbon fluxes immediately after the ice melts because carbon sort of builds up, and there's increased microbial activity in those first few freeze-thaw cycles. And then finally, this also could increase the evaporative window for peatlands as well. Again, sort of signaling that there could be more water losses from these peatlands. So thank you for listening, and happy to take any questions.
Okay, I don't see any questions in the chat or in the Q&A. Andrew, I think, has a very quick question, which we might be able to sneak in while Haley gets her presentation set up. So Andrew, if you could ask your question, and we'll let Haley start getting set up.
Okay, thanks, Brandon. Very nice talk. Quick question for you. So peat is a bit like open water, but also different, right? Because the ice doesn't float in the peat to the top as it's thawing. So I'm wondering if you've looked at the difference in thawing rates between open water and peatlands.
Between open water and peatlands, I have not. I do have thawing rates specifically.
Can you stop sharing your screen, Brandon, quickly? Sorry.
Oh, yep. Nope. I do have the thawing rates for it, but no, I have not compared directly to like open ponded water that would have frozen.
Yeah, okay. Thanks. We better push on, I guess.
Yeah, absolutely. I think most people can get each other's email addresses from a simple Google search of each other's or CGU listservs or whatnot. So, okay, so pleasure to introduce our next speaker. She's one of my students, actually, Haley from Brandon University, using a water balance approach to evaluate peatland restoration in southeastern Manitoba.
Sorry, I'm just having issues sharing my screen. My computer keeps saying that I don't have it so that I can share my screen. I'm just gonna email it to Frank quickly and just do it off his computer. So just give me a second, sorry.
Okay, well, Brandon, I had another quick question while we get that set up then. So you started with completely frozen soil at the beginning of each of your model runs. Mm-hmm. So I'm just thinking, as we get a warmer climate, does that assumption become increasingly invalid? Right? Like, I mean, it would, because you're modeling it, it would be maybe not too difficult. I mean, I'm not a modeler, so you can tell me this is going to be a mountain of work, but, you know, could you start with like a 85%, then a 65% or a 50% moisture content, and then just rerun everything with those initial starting conditions?
Yeah, absolutely. Absolutely. And I have done that. I don't have that for this particular presentation. And what that does is that changes the depth of freezing. So wherever your initial moisture content is, it changes sort of the thickness and depth of your freezing. But interestingly, it doesn't necessarily change the timing so much, because your thermal conductivity is sort of offset accordingly. There's a bit of a negative feedback there, because if you have more liquid water, then you have less thermal conductivity, as opposed to if you just had a straight frozen layer.
Yeah. Okay, I'm going to suggest we actually just move to Frank while Haley gets her stuff sorted out. Haley, if it's in the Dropbox, then I can actually run it off of my computer as well, so we can chat while Frank's going. So Frank, if you want to turn on your camera and share your screen, and we'll get started with Frank. So Frank will be discussing ecohydrology and a slope study at the periphery of a fen restoration. So it's actually the same fen restoration as Haley's site. And Haley, if you want to turn off your camera. As you can tell, they're in the same room.
All right, I will share my screen. Does everyone see my screen?
Yep, we're good, Frank.
Okay. All right, so my research is on ecohydrology and slope study at the periphery of a fen restoration, and it's supervised by Dr. Pete Whittington. So we have to first understand what wetlands are. So according to the National Wetlands Working Group of Canada, wetlands are basically lands that are saturated with water long enough to promote aquatic and then processes and then other biological activities. Now, we have marshes, swamps, fens, bogs, and then the kind of wetlands that we are interested in is peatlands, which basically have their peat being more than 40 centimeters thick. And for the sake of this research, the focus is going to be on the fens and then bogs. Now, why the need to study peatlands at all, and peatlands in Manitoba to be specific? So basically, peatlands act as carbon storage medium, and then they help in mitigating against the adverse effects of climate. And think of peat as also as a source of peatlands, as a source of horticultural peat, basically what you would use to grow your flowers. And then in Manitoba, peatlands are about 38%. There are 38% of Manitoba is wetlands, and about 90% of that is peatland. And so that overall makes peatlands in Manitoba important to study. Now, with the genesis of fens and bogs, we know from the literature that fens are generally fed by groundwater and then precipitation. And then they, over a long period of time, they develop into bogs, which disconnect from the groundwater system and then are fed solely by precipitation. Usually what happens is that this bog peat is usually harvested for peat, which is what I mentioned as used for these horticultural purposes by peat companies. Now also we have to understand what ecotones are in generally. So ecotones are generally biological transitions between ecosystems. Now, if you take, for example, a lake and then you have a forest, you have the transition between the two, and that is an ecotone. And then you have a grassland, you have a forest, and that will be an ecotone, which is a transition between the two. And then for the sake of fens and bogs, we have a fen here, and then we have a tree bog, and then we would classify the transition here as an ecotone. Now, ecotones are very important because of their ability to help in the exchange of materials, helping to enhance the flow of groundwater from one ecosystem to the other. For the sake of fens and bogs, being able to exchange nutrients and then energy. And then based on that, they're able to help to, when there is changes in the ecosystem function, ecotones can help in detecting that change. And then they also have the ability to support biodiversity. Now, for the sake of peat extraction for horticultural peat, we have a peatland, which is functioning properly with high water table. And then peat companies will come in and then drain the water table by establishing ditches. And so that leaves the extracted peatland unable to recuperate. And then this also results in the disconnect between the hydrology of the extracted site and then the surrounding site. So the focus on this research is looking at how we can basically connect the hydrology of the extracted site and then the surrounding site. Now, from the literature, it's stated that blocking ditches and then recontouring is an initial step that helps in resulting in an increment in the water table. Now, basically, you have a cutover peat and then you have a forested peat. So these ditches are blocked, and then you create gradual slopes. And from the literature, that is helpful, or that can help to increase, allow groundwater from the surrounding peatland into the restoration site, and thereby also raise the groundwater table. Now, since ecotones are removed during peat extraction, doing this can also help in the establishment of ecotones. Now, from the literature, ponds and berms have been used to see if we can increase have been used in the literature to understand, to know whether they are significant in increasing the water table on restoration sites. But then in the literature, it's been mainly used for carriver peatland and has not been used on artificial ecotones as a means of raising the water table. And that is where my research comes in. So the hypothesis is that if we creating ponds and berms on the periphery of extracted peatlands during restoration improves hydrological conditions. So the specific objectives is to determine the impact of ponds and berms on water table and water flow, and also to determine the best retention treatments that influences water storage on artificial ecotones. So the research site is in southeastern Manitoba, about 72 kilometers east of Winnipeg, and that is the Moss Per peatland. So the research my research began in 2019. We established 8 transects, and we also have a weather station right here. And then we monitored the water. We installed wells along those transects and then monitored changes in water table. And then we also determined elevation profiles across along those transects. And then we also, in the following year after recontouring, we determined hydraulic conductivities for on various treatments that we applied, which I'll be talking about in the upcoming slides. So with the results and our weather data, for the 2 seasons that we have here, for 2019 and 2020, the total precipitation that we had were below on average below the climate normals for Manitoba, for the site, sorry. But then, as we can see from here, the temperature for the average temperature in 2020 for like July was a bit higher than July 2019, and then both temperature and then for 2020 was also a bit higher than the climate normal. And then for our precipitation, a lot of the precipitation was not really evenly distributed throughout the summer seasons that we did our measurements. We had most of our precipitation for 2019 occurring in July, which was a very heavy rainfall that actually occurred like in a single day. And then in 2022, we had the highest precipitation occurring in June. Now for our elevation profiles, we took 2 specific days. In 2019, we took 2 specific days, one day calling that a wetter day, and then the other day calling that a drier day, and then looked at the difference between the highest and then the lowest water table elevations. And this is for one transect called Southeast. So that is peat here, and that is clay, and we see the maximum and then the minimum water table. So we did that for all the other transects, the east transects, northeast transects, north middle, northwest, west transects, southwest transect, and south middle transect. And then when we compared that data, we saw that the west transect and the east transect had their water tables being higher as compared to the other transects, which was also evidenced based on the vegetation. The west and then the east side had a high abundance of sphagnum moss as compared to the other transect. Now we compare the 2020 and then 2019 data for all the other transects. Now, when we started with recontouring on 3 of the transects, but then, sorry. Now with this graph, we have the water table below, the water level below ground surface, and then this on the horizontal is the transect distance. So for each point represents a well, and then the arrow bars represent the changes in the water table over the period, which was the summer period. And then overall, we saw an increase in the water table for 2020, even though temperatures and then precipitation for 2020 was not as high as 2019. But then we had 3 transects, along which we recontoured, but then we didn't see a huge difference as compared to the other transects that recontouring weren't done on them in terms of the differences in the water tables between the 2 seasons. So as our retention approach, we created ponds and then berms along the ecotone sites. So we have a 5-pond configuration here, and then we have a 3-pond configuration, and then we have berms, and then we also have a control here. Now we installed wells across all these treatments and then we monitored the water table after we had done the recontouring and everything. So there were 3 sites for this experimental designs. We had northwest site, and then the west site, and then the southern site. Now we determined peat depths across all the treatment. And then as we can see from here, the vertical here represents the peat depth, and then the horizontal represents the distance along the transects. And then generally the peat depth for the west side was higher, or basically having like thicker peat as compared to the southern section and then the northwest section. But for each of there wasn't a yeah, I think that was okay. Sorry. Now for the water table measurements that we did for the wells. Now each of the series here, the vertical represents the water table below ground surface, and then the horizontal represents the distance along each of the transects. So, and then the vertical represents the dates. So looking at the treatments that we had, the 5-pond configuration generally had higher water tables, even though there was an overall decline in water table for all the transects. Now we also installed tensiometers to determine water tension, and then the 5-pond configuration also showed higher values for that. Now for hydraulic conductivity across for all of them, we also realized that the 5-pond configuration had higher values in relation to the water table. Now, in summary, the peat depth and the surface elevation, we showed an influence on the groundwater, and then surface recontouring and ditch infilling also improves wetness. And then we also saw that 5-pond configuration maintained relatively high water table. And then we can also see that the ponds and then berms configuration that we applied on these artificial ecotones played a significant role in regulating and maintaining wetness on the constructed slopes. Now, overall, we can see that understanding groundwater flow via these artificial ecotones can improve our understanding in achieving fen restoration goals. Now, I would like to acknowledge the Canadian Sparkling Peat Moss Association and their partners for funding through NSERC, and then to my research committee, Dr. Whittington, Dr. McGonigal, Dr. Maria Struck, and then to those who helped me at the field, Alyssa, Brewer, Vidley, Golubev, Langdon, Baker, Haley, Lobreau, Yona, and thank you.
All right, Frank, thanks very much. I think just given kind of the late start, we'll skip questions so we can see if Haley can get back kind of get us back on track. So Frank, if you want to stop sharing your screen, that would be that'd be great. Apologies to Jim Buttle too. I hadn't actually seen your question in the Q&A, Jim, when I asked basically exactly the same question. So clearly we were thinking along the same lines. So I see Brandon replied formally to you in the Q&A. So apologies for that. I just looked down and I went, didn't I just ask that question? So okay, our next, oh, where to go, Haley?
I wasn't unmuted.
All right, so make sure there's no speaker feedback in the room since you're sharing the room. So using a water balance approach to evaluate peatland restoration in southeastern Manitoba. Go ahead, Haley.
Okay, so to start off, what are wetlands? So wetlands are areas where the water table is at, near, or above the land surface long enough to support wetland vegetation and biological activities. And peatlands are a type of wetland that have accumulated for 40 centimeters or over 40 centimeters of organic sediment. There's two types of peatlands that I'm going to be looking at: bogs, which are ambrogenous, meaning that they're fed solely by rainwater. They get their nutrients from atmospheric deposition and precipitation. They're poor in nutrients and minerals, and they're more acidic. And they're usually dominated by sphagnum, air-cushioned shrubs, and trees. And fens, which are precipitation and groundwater fed, richer in minerals, slightly acidic to alkaline, and dominated by brown mosses and herbaceous plants such as sedges. So why are peatlands important? Peatlands play an important role in the global carbon cycle, which helps regulate the Earth's climate. Their global carbon sinks because of their vegetation production being greater than their decomposition rates, and they store roughly 1/3 of the planet's terrestrial carbon. So understanding peatlands in Manitoba, they cover 11% of Canada's land surface, with 19.2 million hectares of peatlands in Manitoba. 38% of Manitoba's surface areas are wetlands, and 90% of those are peatlands. And extracted peat is useful for horticultural purposes, and the bulk of peat extraction is done on bogs. So understanding the pre-extraction hydrology of a bog. So all bogs were once fens, but not all fens become bogs. So when you harvest a bog, there exists, or in a natural bog peatland, there exists two distinct soil layers: the acrotelm, which is the upper layer, and the catatelm, which is the lower layer. And these layers help control the hydrologic regime. So the acrotelm is responsible for regulation of surface runoff, evaporative losses, and fluctuations in the water table. And the catatelm lacks these self-regulating characteristics. So during extraction, they remove the entire top layer and just leave behind that catatelmic peat. And then these drainage ditches are installed to allow machinery on the field, which helps drain water off the site. This is like the tractors they use to harvest the peat, basically just drive up and down a bunch and vacuum it up. Pre and post restoration, the largest challenge to overcome is the limited availability of water. So during restoration, these drainage ditches are filled in to raise the water table, and a higher water table is the driving force for peatland recovery. So in these photos, you can see like pre-restoration, there's this big drainage ditch, and then post, you can still see where the drainage ditch once was. So my research objectives for my project is, one, to calculate a water balance to quantify how much and where the water is going for future restoration plans, address the influence of peatland disturbances and restoration techniques on snow dynamics, and evaluate the findings in the objective of restoring the site to a fen. So a simple water balance. The ins include precipitation, including rain and snow, groundwater and surface water runoff. Outs are evaporation and runoff, also from surface and groundwater, and change in storage from the saturated zone, which is measured using wells, and the unsaturated zone using moisture probes. The study site is 90 kilometers east of Winnipeg, Manitoba. You can see Winnipeg, and then I put a little star on where it would be located. It's an 80-hectare field site in a subhumid climate. And during 2019, restoration began in 2019, but due to heavy rainfall, it was all put on hold, which gave us a unique opportunity to measure and monitor before and after effects of restoration. So this is a surface view of the finished site. So you can see it's like relatively flat. And then the remnant still has, like, this hasn't been passed with a bulldozer at all. This is just what was left from when they were harvesting. So it still has the drainage ditches and everything. And then the in progress was just one pass with a bulldozer. So it created these little berms across the entire surface. So methods. Each research transect is 76 meters long and consists of the following: a rain gauge to manually measure the rain to see if there was any variability between the different stages of restoration. Lysimeters we installed in groups of 3. These are used to measure evaporation. And then wells, there's 8 per transect. They're all installed to clay, and this helps measure the water table change over time using a blow stick. And then a tensiometer, I installed them 10 centimeters deep, and it measures soil water tension. And then at the weather station, we have a data logger, a temperature and relative humidity sensor, a rain gauge, net radiometer, and moisture probes beneath the surface. During my snow surveys, we did snow depth, which was measured every 8 meters using a metal stick ruler, and snow density, which was measured every 60 meters using a standard Eastern Snow Conference 30 plastic snow tube. This was weighed using this hanging mass scale that Frank's holding, and it was used to calculate snow water equivalents. And we also did snow pits to evaluate the snowpack. So temperature and rain. In 2019, the temperature followed a similar trend to the climatic normals, except in December. You can see it drops lower, and it rained significantly more in 2019. And then in 2020, the temperature data was a little bit warmer in the summer, but cooler in the fall, and it rained less than normal, except in June. Snow depth. The first graph is from 2020. There's only 2 measurements due to COVID. We weren't able to go to the field as much as we wanted to. And in 2021, same thing. But the whited out or open box plot is one date, which was February 18th, March 3rd, and then March 19th, March 8th. And then March 17th is just this one right here in the natural. But as you can see, like, the snow depth was deeper in 2020 compared to 2021, except for in the natural. And here's just a view of like what it looked like on March 3rd here. So we assumed that these berms would hold back more snow, creating a deeper snowpack. But this year, since we didn't have much snow, they actually helped melt the snow faster. And this was what we found on March 8th. And then by March 17th, all the snow was gone from the field site. Snow water equivalence, which is a measurement of how much water will be available during the melt period. Again, we only had one day in 2020 because of COVID, and then March 3rd, 8th, and 17th. Yeah, there was more days in 2021 that actually had 0 for measurements because there was no snow, and it was much more in the in progress. It varied more. For evaporation, I used R script created with the help of Dr. Alex Coiter from BU. So we created the script to calculate evaporation using the weather station data and the lysimeter data that we've collected. So the model refers to the equilibrium evaporation from the weather station, and the actual refers to the lysimeter data collected. And then the alpha value was used to correct the model data, and then the R squared determined the relationship between the actual and model data. So this was just a visual of every single lysimeter that's in the field for the 2020 season. And this is the corrected model evaporation for 2020. It was corrected using averages of the alphas at each stage The in progress stage had the greatest evaporation rate in 2020 with 5.7 millimeters. This is the data from 2020. This red box just outlines a rain event. So it like jumps back up and then descends again. And then this is for 2021 so far. And the in progress stage is showing a similar trend compared to 2020. This is the water table for 2020. Each of these colors represents a different well throughout the transect. This is when the wells start to go dry, when they start to flatten out. So it was in early July. And the finished showed that it held more water for longer, but I believe that's just because the peat was deeper. It still shows that the water table for the site drops too low to support fen vegetation. And I don't have any diagram for 2021 for the water table measurements since all the wells are dry currently. This is soil water tension using the tensiometers in 2020. The remnant stage shows the least variation in pore water pressure, but they all drop below 100 centimeters. And when the pore water pressure falls below 100 centimeters, it's rare for sphagnum recolonization. So putting this all together, so the ends precipitation, rain was measured by the weather stations and the manual measurements, and snow was measured using the snow water equivalents. Groundwater and surface water was actually measured by Frank Yamoah, the student who presented before me. The outs was evapotranspiration using data collected from the lysimeters, comparing it to the data collected from the weather station, and then calculating using the Priestley-Taylor method through the R script. And then runoff was not directly measured, but the flow measurements conducted at the outflow drainage ditch were roughly 0.054 millimeters a day. So very, very few, very, very little. Change in storage of the saturated zone was measured using the change in water table depth from the wells. And the unsaturated zone was measured using moisture probes at the weather stations. So preliminary findings for my first objective was calculating the water balance to quantify how much and where the water is going for future restoration plans. Since we found that evaporation is the dominant water loss from the site, and the site average evaporation rate was 4.8 millimeters a day from May to August in 2020, which is quite a lot when there's no rain. And my second objective was to address the influence of peatland disturbances and restoration techniques on snow dynamics. Based on the limited data collected, any sort of recontouring helped accumulate more snow, which leads to a deeper snowpack and a larger snow water equivalent value, which helps insulate the ground better, leading to a decrease in soil frost and promotes spring snowmelt to infiltrate rather than run off the surface. And objective 3, evaluate the findings and objectives of restoring the site to a fen. Driven by evaporation rates, even the finished site dried too much to support fen vegetation. And since we found that evaporation is the dominant water loss, a straw mulch will be applied to help reduce evaporative losses. So future plans, this is in the works. It should be happening within the next week or so. We're going to block the drainage network to hopefully raise the water table and install an outflow drain at the drainage site I was doing my outflow measurements from, just in case there's a surplus of water. And then we're going to put down straw mulch and a new research transect to see if evaporative losses and water table drawdown changes. So I would like to acknowledge the Canadian Sphagnum Peat Moss Association and their partnership with NSERC for helping sponsor this research financially. My research committee, Dr. Pete Whittington, Dr. Alex Coiter, Dr. Pamela Rutherford, and Dr. Scott Ketcherson for all of their help, and to the field help from Alyssa, Yana, Frank, Lyndon, and Vitaly. Here's my references, and yeah, done.
Excellent. Thanks, Haley. If we're sticking to a roughly 5-minute late schedule, then there might be just enough time for one quick question if anybody has one. If not, then I'll just get started. I don't see any in the Q&A, so I think we're probably good just to try to keep on keep on track. All right, share screen 2. There we go. Okay, so I'll just get started and introduce myself. I should really introduce the first author, Vitaly, who was my master's student a couple of years ago. So we're kind of finishing off publishing the rest of his thesis. So he's first author on this paper. We submitted this to Journal of Hydrology a couple of months ago. It's been accepted with major revisions, and we'll be resubmitting it back to the journal shortly. So if you're interested in this, it should be published shortly. And then I would imagine a lot of people on this call probably know Colin as well, who we kind of brought in after the supervision aspect of the thesis, because he had some really good insights to this. So what? Who cares? So we know peatlands store a lot of carbon, and most of that is by sphagnum, and we know biogeochemistry is largely controlled by hydrology, and therefore the hydrology of the sphagnum is largely controlled by the soil hydraulic properties, so the soil water retention curves, at least in the unsaturated portion of the profile. So we can get those from the Van Gunukten Malum model in RET C. So there's just a screen capture from RET C there. We also noted that, almost all of the people, and they're all come out of Price's lab, so it's perhaps not surprising that we pretty much just always used 5 centimeter high samples. But is that the best? Could you use a larger sample to preserve some of the poor connectivity throughout the sample to get a better representative sample? And so do the soil hydraulic properties vary between species, and does it matter ecohydrologically? So those were sort of the objectives of Vitaly's thesis. So that's Vitaly there at one of the field sites in southeastern Manitoba, taking a big monolith of soil, which we then cut up into 15 centimeter high samples like this. So we had 8 times for Sphagnum fuscum, which is a predominant hummock forming species, versus Magellanicum, which is a more generalist species. And it's really important to note that Magellanicum no longer exists in Canada. There was a paper, the Hassel et al., that just came out. I hadn't actually heard of this, but one of the reviewers pointed it out. And so it's almost certain that the Magellanicum is actually Sphagnum divinium, based on some other samples that we've taken more recently. So anytime I say Magellanicum, I really should be saying divinium, but the general process is that as identified, it was Magellanicum at the time. So we then take that sample and sandwich it between 2 tension discs that had the same pressure head. So there was a uniform pressure head throughout the sample, and therefore the flow was just driven by the gravitational component of hydraulic head. There it is there in the lab with the 4. We were able to run 4 of these at once, the 15 centimeter cores. We then split those up into 3 cores, a top, middle, and bottom. So you can start to quickly see that the number of samples ramps up pretty quickly. Vitaly automated this whole system with solenoid valves that would open and close based on water completing the electrical circuit between here, all into microcomputers. And I mean, it was amazing to see him build this, because I mean, this is probably 2,000 to 3,000 hours of work over the course of a year to have this sort of automated system. So what do we find? So I'll show you a couple of box plots. So the filled is fuscum and the open is Magellanicum. We have volumetric water content here on the right. Note this might break to get up into the saturated conditions with 0 pressure head. And then we have, so each one of these box plots would be n equals 8. So 8 tops blue like the sky, 8 middles green like the earth, and then 8 bottoms, the core red of the earth, if you will. And then the gray, which I'm sure if you just mashed all these colors together would be kind of a gray mess. So what we can see if we add the rest of the box plots in and basically connect the lines is that this is just a soil water retention curve if we basically plot through the medians. So you can kind of see that with the open symbols for Magellanicum, there is a bit of overlap between a few of the layers, and that the overlap becomes a little bit more as you go down. If we looked at the difference between the top and the bottom for Fuscum, we can see that the bottom had much higher, probably about 20% more retention at any given pressure step, denoted along the bottom here, and then kind of broken up with the vertical dashed lines. But this was actually the opposite up here. This wouldn't be retention. This would actually just be equal to the porosity because it's saturated. So the top had a higher porosity than the bottom, which you'd probably expect. So the key points here is that water retention increased with depth within a species, and that the 15 centimeter core retained less water compared to sort of taking the average of the three of these. And we think this, and you know, it's kind of embarrassing once you think about it after the fact, that you probably could have figured this out, but it would be very difficult to maintain a 5 centimeter pressure head across a 15 centimeter sample. It would just drain out because of the gravitational component of the sample. And so what we sort of have figured, and the reason that these differences are more pronounced at the lower pressures, sorry, at the higher pressures closer to zero, is the core was just draining. The tension discs weren't able to hold that moisture in the core, in the 15 centimeter core. So if we switch up and go to hydraulic conductivity, fast at the top, getting slower as we go, then we can see the same sort of K theta relationship as well. And then the variability with our sphagnum fuscum top being quite a bit different than the other fuscum. So looking just at the filled symbols, and yet the Magellanicum seems to be, you know, kind of overlappy, but there's a fair bit of overlap between them all. So some key points here, K unsat increased with depth. And so we see that at the bottom, the hydraulic conductivity is higher than at the top. And the opposite is true, the saturated hydraulic conductivity with zero pressure up here. And so if we look at the K theta relationships, which ultimately kind of controls for some of the variability there, and we plot those, we were concerned that these points were kind of anchoring and overinflating the value, if you will, of the, like the R squared, like overinflating the significance of the relationship. So we replotted them with everything under saturation. So it's this plot here on the right is literally just these data here. This is moisture content along the bottom and hydraulic conductivity up the top. And then we add in the 15 centimeter data overlaid. So what we did is we took the lines from the top and just put them grayed out in the bottom and removed the points. So you can see that the 15, you know, similar slopes, if you will. But looking at these and doing an ANCOVA, we actually find that most of these slopes are statistically significantly different from each other, denoted by bold if it's 0.05, and if it's bold and gray background, then 0.001 significance. So quite a few of them were statistically significantly different than each other. So who cares? Why does this all matter? So this is where we've kind of brought Colin in to help us with some hydrological modeling. So we did a 30-day drought period using Hydrus 1D, using locally available data, actually from a field site very close to Haley, and using some of Haley's data. We modeled a 15 centimeter core with 100 nodes. When we put in the 15 centimeter data, there was just one soil material versus the 3 soil materials for the 3x5. We had an evaporation rate of 2.6 millimeters per day, which kind of shut down overnight and then picked up, you know, around 2 or 3 in the afternoon, I think it was, in the sinusoidal pattern. But sort of importantly is that our initial boundary conditions for the bottom were based on their landscape position. So we started Fuscum 25 centimeters higher, or put another way, a minus 52, 53 centimeter water table than Magellanicum, which we started with a minus 27 centimeter water table, which was reflective of their position in the landscape and the hummock hollow topography in the bogs in this area, about 25 centimeter difference, which we thought better simulated this hummock hollow topography. And then we got all of the parameters from curve fitting in RET-C. I can get into more details on that later if you'd like. So looking at the first time step, so time step 0, you don't see anything. And then 6 hours later, we have our 1/15 with the solid line, so that's intact, and our 3/5 as the dotted lines because it's broken up, because we cut it up, if you will. So we'll just basically zoom to all of the data. So it's the same y-axis throughout, 0, 5, 10, 15 centimeters, and then we get down to 6, 15, and 30 days. And so you can see the Magellanicum doesn't really do anything different between the types of models, between the 1/15 and the 3/5. And the middle line, the grayed out solid line, is data where we got the RET-C coefficients without including any of the 5 or 10 centimeter pressure head data. We were curious if we could just throw those out and, you know, what the results basically would look like. So we can see certainly that Fuscum is getting more stressed, and that the 1/15 centimeter shows the most stress. This black line out here is always further out to the left, so a higher pressure head, if you will, or a lower pressure head, sorry, higher tension, more negative. So that we sort of speculated that this was because of the sort of poorly represented upper layer when you have a 15 centimeter sample between these tension discs. So we basically concluded that it doesn't make sense to use 15 centimeter samples, at least in the apparatus as sort of described in the couple of like Price and McCarter papers over the years. So just to reiterate, Magellanicum, not much variation and never really stressed. If we define stressed as sort of being over minus 100 centimeters, which is some Price work from Quebec earlier on in the kind of late 90s, early 2000s, then this magic number of about 100 centimeters there. And so for both, it's important to point out that neither of them exceeded this minus 400 centimeters. So we wanted to know basically what the water budget of the moss was. And so we took the difference between the ins and the outs. So the outs being the top, the evaporative flux, and then the soil water flux coming up from the water table would be the influx, the bottom flux. And we can see Magellanicum there in red lost a lot more water, or was able to funnel more water through the system than the Fuscum, which lost maybe 4 or 5 millimeters versus Fuscum's almost 11. And you can see the diurnal variations there. And this weird kind of dip here at the bottom is just the model equilibrating basically from time 0 and the moisture settling out and basically draining out the bottom. So the key points from this: losses are limited in Fuscum. Its lower hydraulic conductivity at those moisture contents kind of prevents it from losing that moisture. And that it is very important to represent the ecological niche that these species rest in the atmosphere, in the environment where they actually exist. And so, you know, an easy way to do that is with water table, but you can very easily change things with a model and manipulate the water table. So if we take that graph and add a few more lines, and this gets very confusing to look at, so try not to read ahead too much, but the 2 lines there that I have indicated with the blue was the original data from the previous graph that I showed you there. But if we just compare those 2 lines, then that is Fuscum with Magellanicum's water table, and then the red line is Magellanicum with Fuscum's water table. So doing a comparison, we can see that here, when everything is correct, we have red on top of black, or red losing more than black. But when we swap the water tables, all of a sudden Fuscum is now the one losing a lot more water, and Magellanicum is losing less water. We also have the advantage that if we compare between the colors, so Magellanicum with its correct water table, Magellanicum water table, compared to Fuscum with the Magellanicum water table. So we can kind of compare the effect of water table location. And so here would be, again, the different species, Magellanicum and Fuscum, but with the same water table, the same Fuscum. And so this is actually what Colin did in one of his papers a few years ago, which we would argue is incorrect now. If you're not taking into account landscape position, then you're kind of comparing these two and coming up with your results, when really you should be comparing the blue line with correct water table positions in the landscape. So take-home messages, you can read them there, but soil hydrologic properties do vary between species. The tall samples have experimental limitations. The soil hydraulic properties seem to be consistent with their landscape position, and we need to consider the ecological setting when modeling these. And so I'll just leave the thanks up there, and I'll moderate any questions that anybody might have. Don't see any in the Q&A. Also, the attendees are now dropping off, so we're...
Can I ask a question, Pete?
Yeah, of course, Andrew.
Just a quick one.
Yep, absolutely. And then we'll watch the YouTube video together afterwards.
Can you hear me?
Yeah, go ahead, Andrew.
Sorry. Yeah, how did you measure hydraulic conductivity as a function of saturation? I don't know if you said that, but I missed it.
We didn't measure it directly as a function of saturation. What we did was we were able to get the hydraulic conductivity at different levels of saturation. So we never actually did a soil water retention experiment. We got the moisture content from the mass of the sample when the outflow had equilibrated, once we had a constant outflow. And so we then knew the moisture content and the K at that like intersection of those two values, basically, which then we just plotted by taking the moisture content and the K that it gave us.
And how did you get the K? Sorry, I missed that.
So Darcy's law, basically. So with this, you have the you have a pressure gradient across here. So you have a change in head. You have the area of the sample.
You're
and then
not
you have
sharing your screen if you're.
Oh,
pointing.
sorry. Yeah, that's right. Can you see this now?
Yes, yes.
Sharing the right screen. So you have a pressure gradient across here. So you have a change in head from top to bottom. You have a length. So that's dH by dL for Darcy's law.
Yeah.
You have outflow. So capital
Mhm.
Q there. And then you have the area of the sample. So Darcy's law is Q equals K dH by dL times A. So just rearrange to solve for K. So that's Q divided by A multiplied by dL by dH.
Okay, thanks. That's good.
Yeah, no problem. Okay, so thank you to our attendees for kind of staying till the end, and we'll just watch a quick video, which I think Oshua will show us. Excellent. Thank you for that. It's a great video. So I do encourage you to continue joining our seminar series, as well as the AGMs that we have coming up. So look for emails about that. And so whatever time it is, I think it's afternoon everywhere across the country now. So have a great afternoon or early evening, depending if you're way out on the East Coast. Have a good day, everybody.